Right. Good morning, boys and Pallavi. Today, again, we are back with the same chapter, Cell, Unit of Life. And to overall, comprehensively, we were talking about what is a cell, types of cells, and the various cell organelles that are present inside the cell. And in that, we were talking about important concept called fluid mosaic model of the plasma membrane, and we were also discussing about the nicknames that we were giving to various cell organelles such as mitochondria. In yesterday's session I said are called powerhouses of the cell. Similarly, lysosomes, suicidal bags of the cell. Ribosomes, protein synthesizing factories. And today's session, plastids, kitchens of the cell. Adikimane, they are kitchens of the cell. Why are they so called? As you can guess, plastids are responsible for preparation of plants' food. What is plants' food? Starch. Starch is the food of the plant, as all of us know. And also, you know, biochemically, starch is a carbohydrate. Just like all sugars are carbohydrates. Rice that you are eating is a carbohydrate. Bread, chapati, puri, all these are carbohydrates. So today, when you are having your breakfast, lunch, dinner, just observe what is that you are eating. South Indians, we are predominantly carbohydrate eaters. Just observe it, including your kadak roti. They're all carbohydrates, right? Whereas, if you see it move towards the north, their diet will be predominantly protein oriented and fat oriented. And that is the reason the scientists and the nutritionists always advise us to have a balanced diet. We should have everything. Carbohydrates we should have, fats we should have, proteins, minerals, vitamins or vitamins, and of course, I have been always advocating you should have 6 to 8 liters of water in a day. You must be always hydrous. Okay? Drink plenty of water. But precaution is good water, good quality water. Don't drink dirty water. And being biology student, you know 90% of the diseases are waterborne. So if you take care of water and the air that you breathe in, you are healthy. And the third most important is the food that you eat. Just focus on that, then everything will be all right. You are a healthy boy, you are a healthy girl, right? So the plastids basically, they are, as I said, food factories, the kitchens of the cell. And based on the pigment that is present in plastids, coloring substance. Pigment means coloring substance. Plastids are of 3 types. Chloroplasts, which all of us are familiar, green in color. And they are green in color because of presence of a green colored pigment called chlorophyll, right? And second one is chromoplast. It is responsible for varieties of colors like yellow color, brown color, red color, orange color. These are all because of chromoplast. And the third one is leucoplast. It is colorless. Out of the 3 plastids, leucoplast is colorless. And if it is colorless, what happens? What does it do? It will make the food to be stored. So it is a storage organ. Just like our food will be stored in stomach, like that, leucoplast will store the food material, right? So the chloroplasts are predominantly present in all the green plants, as all of you are seeing every day, isn't it? So chloroplasts contain chlorophyll, a green colored pigment. But this you know already in junior classes. But here you will come to know that when you come to school and perform one experiment called paper chromatography, wherein you can see these various colored pigments virtually running on a paper with different colors. That's why we call it as paper chromatography. There you can see even in the chlorophyll, varieties of chlorophylls are there. Chlorophyll A, chlorophyll B, like that. Okay? And we have got xanthophyll, carotenoids. These are all pigments that are responsible for different colors. Right? Right. So, second one we said chromoplasts. These are contain fat soluble carotenoid pigments like keratin, xanthophyll. This gives yellow, orange or red color. And leucoplasts, just now I said, colorless plastids of varied shapes and size, and they are mainly responsible for storage of food. So they include three types among the leucoplasts, colorless plastids. Three types are there. Amyloplasts, they are mainly store starch, like in potato. Eleoplasts, they store oils and fats, like in many seeds you must have observed, right? And alleroplasts, they store proteins. So they are colorless, but they perform the duty of storage. And now we focus on the green colored plastid, that is chloroplast. And a chloroplast basically will have two membranes, an outer membrane and inner membrane, just like in mitochondria. Okay? And they are, where do you see them in the plants? Like if you take the section, again you will be doing this experiment when you come back to school. You take the leaf and take the TS, transfer section of leaf. When you take the TS, you will see two types of cells. Upper layer consisting of pillar-like cells, and bottom layers will be having amoeboid shaped cells. These are called mesophyll cells. In that, there will be lot of chlorophyll. And what is the shape of this chloroplast? Normally it is oval shaped or spherical, discoid, and sometimes ribbon-like also. And the length 5 to 10 nanometers, width 2 to 4 nanometers. The numbers vary to 20 to 40 cells in the mesophyll. Now here, there is a lot of research going on this. Some scientists are getting an idea. You know, always I tell you, think out of hat, think out of box. If 20 people are there and one question is posed to the 20 people, everybody thinks in the same way, then there is no uniqueness. Out of the 20, 19 think in one way, and you think, being Sunday school boy, think different. You are unique, and you are selected. That's what happens in SSB, right? So some scientists started thinking out of hat. They got that, I should say crazy idea. If within a cell 20 to 40 chloroplasts are there, how about increasing them? Let's say we make it some 80 or 100. What will happen, Naveen? Pallavi? Yes. What is the responsibility? Chloroplast's duty is to perform photosynthesis. And what is the end product of photosynthesis? Starch formation. Remember, you were writing in junior classes, CO2 plus H2O in the presence of sunlight and chlorophyll. Look at that, you were writing chlorophyll. Where is that chlorophyll? Here, in the chloroplast. So if you increase, double the number of chloroplasts, automatically chlorophyll also will be doubled up. And what is the role of chlorophyll? It absorbs sunlight. Remember in your 10th class, you studied a chapter called Life Process 1 and 2. In that, I am sure you must have discussed light reaction and dark reaction, Hillman reaction and Darkman reaction, Blackman reaction, right? Blackman reaction. Do you remember that? So basically, the idea is to increase the carbohydrate content, starch content. And if a food is more, the plants also will be nice. They will be flourishing. Now, if you see under microscope, how does a chloroplast look like? This is how it looks like. We have taken the section of it, right? Basically, it is oval shaped. So, as I said, there will be two layers. One is the outer membrane, as you are seeing, another is the inner membrane. And inside, as usual, there will be matrix, right? Stroma or matrix. And then you can see just like 1 rupee coins. You take 1 rupee coins and arrange them one above the other in a row. We say stack, S-T-A-L-K, stack of coins. Exactly this granum look like that. So that each 1 rupee coin you are arranging is called thylakoid. Okay? And many thylakoids, if they are arranged like 1 rupee coins and put them there, then it is called granum. And you can see that 1 rupee coins row, many rows are there. Then plural, grana. I hope you are following me. So basically, plastids are of 3 types. Out of them, green colored plastid is called chloroplast. These are seen in all the green parts of the plants. And when we take the section of that green part, inside there is a tissue called mesophyll tissue. Inside the mesophyll tissue, the chloroplast will be there. And when we take the section of chloroplast, you will see these structures. What is that? Basically, a chloroplast has 2 layers, outer membrane, inner membrane, right? Outer membrane, as usual, it is for protection. Inner membrane is forming, you see, small thread-like structures connecting that 1 rupee coins. Okay, someone is waiting. Okay. Right. So, it is basically 2 membranous structure, outer membrane, inner membrane. And inside there is a vacuum gap with some fluids. We called in mitochondria matrix. Here we are calling it as stroma. What ma? Stroma. And in the stroma you can see that inner membrane is connecting those 1 rupee coins. They are called lamellae. Lamella or lamellae. And how this granum will be, just like if you take some 1 rupee coins, arrange them one above the other, you get a row or stack of coins. Exactly, the granum would look like that. And each 1 rupee coin, round disc-shaped structure, is called thylakoid. Why are we studying this anatomy of chloroplast? We want to show and observe ourselves where exactly this chlorophyll is there. We said chlorophyll is present in chloroplast. Chloroplast is present in mesophyll tissue. Mesophyll is present in green parts, isn't it? In chloroplast, where exactly? It is in the thylakoid. And what is a thylakoid? A small 1 rupee coin size shaped structure. And many grana will be there inside. Right? So this is the structure of a chloroplast. Now let us see the notes of it. So basically, inner membrane of chloroplast is less permeable. The space limited by the inner membrane of the chloroplast is called stroma. It contains number of organized flat membranous sacs called thylakoids, that 1 rupee coins, okay, thylakoids. And the membrane thylakoids encloses a space called lumen, and chlorophyll pigments are present in thylakoids. This is what our intention is when we are studying anatomy of the chloroplast. We would like to pin where exactly the chlorophyll is present. So it is in the thylakoids. So thylakoids are arranged in stacks called grana. Just remember grana. Just like 1 rupee coins you arrange one above the other, how does it look like? Same, the grana will look like. So these are flat membranous tubules called stroma lamellae connecting the thylakoids of different grana. One grana is connected to, one granum is connected to another granum through this lamellae, and lamellae is the extension of inner membrane. So stroma contains small double-stranded circular DNA molecules. Now see, just like mitochondria, chloroplasts also, they have their own DNA. This is again unique feature of the chloroplast. That means a cell will have its own DNA in the nucleus. That all of us know. So there is basically a cell, living cell. Inside the living cell, there is a liquid called cytoplasm. Inside the cytoplasm, all these cell organelles that we are talking for the last 3 days will be floating, including the nucleus. Inside the nucleus, again one more liquid will be there, that is nucleoplasm. And inside the nucleoplasm, chromosomes will be there. Inside the chromosomes, DNA will be there. So this is about the cell DNA. Along with that, 2 other organelles are having their own DNA inside. One is the mitochondria, second is the chloroplast. So many times in your UPSC ND exam, this question will be asked. Name a cell organelle that has its own DNA. There you must be careful. What is that? A, you will give something, endoplasmic reticulum. B, you will give mitochondria. C, he will give some other thing like vacuole. D, you will say A and C. So you have to see if both are given, mitochondria as well as chloroplast, and there is option where both are included, that you must opt. You should not opt either mitochondria or chloroplast. Take care of that. See, when your most important thing is focus on reading the question. You must not skip some important words, right? And in the alternatives, if only either chloroplast or mitochondria is there, okay, no problem, you go for it. But both are there, then you have to answer for both. Like he will give you both A and C, both A and D, like that. Go for that, right? So stroma contains small double-stranded circular DNA molecules, ribosomes and enzymes for the synthesis of carbohydrates and proteins. And all of us already know ribosomes of the chloroplasts are smaller than the cytoplasmic ribosomes. We will be studying about that ribosomes next. And look at that. This is how it will be. So you can see the stroma, thylakoid lumen. Lumen is inner space. Granum, singular, stack of thylakoids. Inner membrane, outer membrane, and the space present in between inner membrane and outer membrane. Okay, Kiran is waiting now. Okay, right. Let me see whether room is unlocked. Right. So next one we would go is ribosomes. Already we said ribosomes are round spherical structures present in the cytoplasm. And remember from your memory, yesterday when we were talking about RER, what is that? Rough endoplasmic reticulum. Remember? And that endoplasmic reticulum has become rough because of presence of ribosomes, isn't it? And I said ribosomes are protein synthesizing factories, and ribosomes will have two parts, one big part, one small part. And thanks to this gentleman whose photo you are seeing, George Pellet, first observed ribosomes. Again, UPSC NDA question. Contribution of scientists towards the development of science. There, many scientist names will come. You have to remember that. So, ribosomes, non-membranous granular structures composed of RNA and proteins. So, ribosomes will be having RNA. And George Pellet in 1953 observed ribosomes first time. Eukaryotic ribosomes has two subunits, 60S and 40S, and they together form 80S ribosomes. I told you already, do not think in terms of mathematics. Bigger one is 60S, smaller one is 40, 60 plus 40, 100, but it is becoming 80S. Why? It is only density, in terms of density we are speaking, not the numericals. So this is how the ribosomes will be there. Let me expand this. See, there are in two places ribosomes are present. Basically, they are round globular structures, just like air bubbles. They will be. And they are spread across the cytoplasm. So they are called free ribosomes. And some ribosomes are associated with endoplasmic reticulum. On the surface of endoplasmic reticulum, ribosomes are attached. Because of their presence, the surface of the endoplasmic reticulum becomes rough. Hence, it is appropriately called as RER, rough endoplasmic reticulum. And I said that ribosomes have got two subunits, one small unit, one big unit. And in all the eukaryotic animals, including us, you see, ribosomes have got one big unit, 60S, one small unit, 40S, and together it is making a 80S component. And what is the function of these ribosomes? Already we said they are responsible for protein synthesis. Okay. Next we move to cytoskeletal structures. Cytology, study of cell. Cytoskeleton means what the cell is made up of. So it is a network of filamentous, proteinaceous structures present in the cytoplasm. Inside the cytoplasm, lot of fibers will be there. Thread-like structures, hagga antivala, thread-like structures. And how do they help the cell being present? They provide mechanical support, motility means movement, maintenance of shape of the cell. Three things they are doing. One is they are giving mechanical support. Second, they help in movement of materials. Third, they give shape to the cell. Look at that cytoskeletal structures, how they are. This is how they are. These are all the various fibers, hair-like structures present there. Consider. And most important out of those are two structures called cilia and flagella. Remember paramecium? I hope all of you have seen under microscope paramecium. On the outer surface, there will be hair-like outgrowths. These are called cilia. In paramecium, you must have seen hair-like structures present all around the body. On the other hand, flagella, they are whip-like structures. Whip, big thread-like structures. So, cilia are small structures which work like O's and causes the movement of cell or surrounding the fluid. What is this O's? In Kerala, every year there will be boat racing. Just like you have got that Formula 1 car racing, like that in Kerala, boat racing will be there. And the people sitting in the boat, they will be using one wooden structure with which they push the water current and the boat goes forward. That's hose. So here cilia also will be doing same thing. They will be making the movement in one way, push the organism like paramecium ahead. And flagella on the other hand is a longer one, I said whip-like, and it helps in cell movement. And flagella prokaryotes and eukaryotes are different together. In what way? Look at that. This is the cilia flagella basic structure. So there will be a basal body at the base, then lot of smaller threads will be there. Okay, we call it as transition fibers, and then inside when you take the section of the each fiber, that you have got different structure. They are called axoneme. So when we make this bigger, look at that, it appears like this. Okay. Now cilium and flagellum. Flagellum singular, flagella plural, right? Cilium singular, cilia plural, are covered with plasma membrane, and their core, that is axoneme, has many microtubules running parallel to the long axis. Just now you have seen. And the axoneme has 9 pairs of doublets of radially arranged peripheral microtubules and a pair of central microtubules. This is called 9+2 array. Now this is what exactly we are showing. Just in the picture you please count how many doublets are there. 1, 2, 3, 4, 5, 6, 7, 8, 9. And centrally how many? Single ones, 2 are there. So it is called 9+2 arrangement. Normally this question also will be there in your MCQ. The structure of axoneme will have in cilia and flagella will have 9+2 array arrangement, 10+2 array arrangement, 8+1 arrangement, like that. Okay. So basically if you see, the outer membrane is plasma membrane and the microtubules will be outside. Periphery means outside, and in between the microtubule doublets there is smaller globules. They are interdouble bridges, and of course central microtubule, 2 will be there, pair, and then there is a radial spoke, and all this is covered by a sheath called central sheath. Again, today after my class, please draw the diagram, practice this, label this, and remember, in all the competitive exams this question will be there, this 9+2 arrangement, right? So basically it is the axoneme structure. So the central tubules are connected by bridges and are enclosed by central sheath, as I have shown just now. It is connected to one of the tubules of each peripheral doublet by a radial spoke. That is the importance of radial spoke. Thus, there are 9 radial spokes. Let us see the radial spoke. Are they connected? And there are 9. See, radial spokes are 9, and they are connected to central sheath. Observe the diagram. You can see. And then the peripheral doublets are also interconnected by linkers. Now see the linkers. The outer ones, they are interconnected by the linkers, interdoublet bridge. See? Those are linkers. And then cilium and flagellum emerge from centriole like structure called basal body. And this is what we expand. Basically we see the basal body, and when we expand the basal body you will see this 9+2 arrangement. Okay. And next we go to centrosome. Centrosome is an organelle containing 2 non-membrane bound cylindrical structures called centrioles. Like in the cell there will be organelles called centrioles, and these centrioles help in cell division. During mitosis, meiosis, centrioles will pull the chromosomes to opposite poles, and these centrioles are present in a structure called centrosome, and they are also surrounded by pericentriolar materials. The centrioles lie perpendicular to each other. One will be present up, one will be present down. We say opposite poles. They are made up of 9 evenly spaced peripheral fibrils of tubulin. Tubulin is a protein, okay? And each of the peripheral fibril is a triplet. The adjacent triplets are also linked. Now let us see the structure of centrosome. Look at that. So basically, centrosome will be present in the cytoplasm. Now let me expand this. Yes. Look at that. See the presence of centrosome inside the cytoplasm, and we are expanding that centrosome. So centrosome has got this pipelines, fibers in the form of pipelines. They are called centrioles. And how the arrangement? These centrioles, just like you have seen flagella and cilium, they are also present in the form of triplets, and there are totally 9 structures in a bundle, 9 evenly spaced, we said, okay? And then we expand further. The central part of centriole also is made up of proteins and is called the hub, which is connected with tubules of peripheral triplets by radial spokes made up of protein. The centrioles form the basal body of cilia or flagella and spindle fibers that give rise to spindle apparatus during cell division in animal cells. Now look at the structures. Yes. So we have got that the centrioles, and in between there is interconnecting materials, fibers, and you have got proximal ends, and there are 2 mother centrioles and daughter centrioles. What is important at present, you must remember that these are arranged in the form of triplets. What are arranged in the form of triplet? What do you mean by triplet? 3 structures. Like that, how many? 10 are there. Means 10 into 3, 30. And the triplet is one microtubule. Microtubule, like you must be using in chemistry lab, that pipette like structure, small tube, glass tube, like that. So what is the function of this centrioles? I said they are responsible for cell division. That much you must remember. They are responsible for cell division. Okay? Right. So these are the 2 structures that we talk of. And then we come to nucleus. Perhaps the nucleus already we started studying it. We know a lot about this nucleus, but still we must be thankful to this gentleman. Who is that? Robert Brown. So he was responsible for the discovery of the nucleus, and he described it way back 1831. And based on presence and absence of nucleus, remember, we divided the cells into 2 categories, prokaryotic and eukaryotic. Remember that? If there is a clear-cut nucleus present in the cell with a prominent nuclear membrane having all the genetic material like DNA, RNA, we said eukaryotic cell. And animals having eukaryotic cells will be eukaryotes. And the other hand, if nucleus is not prominent, nuclear membrane is not there, DNA, RNA has spread, then we say prokaryotic, primitive. We called it as nucleoid. Remember? So the nuclear material is basically stained by the basic dyes. It was given the name chromatin by Fleming. Normally, a cell has only one nucleus, but some cells have more than one nucleus, and some mature cells lack nucleus. Now, here I must tell you one important word. If a cell has got normally one, but some cells will have more than one, then we say xenocytic condition. That condition is called xenocytic condition. And remember, RBC is enucleated cell in our body. Enucleated, no nucleus. Okay? Mammalian RBC has no nucleus. And CU tubes in the vascular plants in phloem, there will be no nucleus. You must remember this. And nucleus contains, as we said, nuclear membrane outside, chromatin reticulum, nucleolus, and of course nucleoplasm. Now look at that. This is how the nucleus will be. Let me expand that. Yes. See, there are two: outer membrane, inner membrane, and then there is presence of DNA, ribosomes, nuclear lamellae. So these are the various structures that are present in the nucleus. And why should we study about nucleus? Already I told you, nucleus is very, very important. It is command and controller of the cell. Whatever happens in the cell, it is because of the presence of nucleus. And without nucleus, cell would be having a lot of problems. In the absence of a controller and commander, you can imagine how the organization will be. Similar will be the fate of the cell. So, in our next session, we will be studying in detail about the nucleus, the various structures that are associated with the nucleus. Until then, take care. You need to draw two diagrams and put a thumbs up in our WhatsApp group today. Diagram number 1 is mitochondria. Diagram number 2 is chloroplast. Okay? Take care.